Welcome back, friends. In this video tutorial we'll be talking about a Bacteriophage lambda, and we'll be talking about different aspects of Bacteriophage lambda, the structure of Bacteriophage lambda, the genome orientation and genome structure, and different genes that are responsible for converting the lambda lytic and lysogenic cycle, which is very important topic. Okay, now previously we have already talked about that lytic lysogenic switching, so in this video we'll be not talking about that. We'll be talking about how the lytic cycle actually established and how lysogenic cycle. So let's begin with the structure. Now lambda is a bacteriophage because it infects a bacteria, right? So it's a virus that infects bacteria. Now in this lambda is belonging to the family Siphoviridae because their structure resembling the structure like siphon or siphon, we call them the Siphoviridae. So let me take the color here. Okay, so we call them Siphoviridae, and the host for this lambda phage is K12 strain of Escherichia coli. Now E. coli are having many different strains. Among them K12 is very important strain, and this lambda is infecting those K12 strain of E. coli. Now if we look at the structure of lambda phage, what we can see here, this is the electron micrograph. This is also electron micrograph of lambda phage. Now in this case we can see that there are three important regions for this lambda. One is this head, so this is the head region of lambda phage. Second one is the tail region of lambda phage, and third is called the J chain at the end, right? It's called the attachment fiber or tail fiber. It's called the tail fiber, or simply called as J chain of lambda. Now this is the chain which is important. It will something look like this. So if I draw it schematically, how it will be represented, it will be represented like that. So let's say that this is, and the J chain will something look like this. Okay, and then the icosahedral head will be aligned like that. Okay, so that's how the lambda actually looks like. And the lambda phage is not having any kind of base plates or spikes and all these things that you can see in many of the images that you're going to find in your book and internet, because those type of phages are different. Those are called usually T4 phages and other type of phages, but lambda phages are not having those base plate or that kind of spikes. In turn, they are having a kind of tail fiber which is required for their attachment, right, or adherence to the host cell. And they are having this kind of contractile tail, non-contractile tail, sorry. Now there is a difference between this lambda phage and T4 phage. If I am talking about in my YouTube channel, you can find videos and structure about both lambda phage as well as T4 phage. And you can find this in T4 phage. What we can see, that tail region is kind of contractile and it behaves like injection. So this tail region, if this is the length of the tail, it can be contracted to this small amount, and due to this contraction they can inject the DNA or genetic material inside the host cell, right? Now but in case of lambda phage, the tail that they are made up of, this is made up with non-contractile rings. Okay, so they cannot contract, but instead what they can do is that they can push the DNA inside, and but this tail region is a little bit longer in case of this lambda, whereas in case of T4 this tail region is a little bit shorter. Okay, now the lambda tail region is little bit more flexible in nature. So you can see in these images they are kind of bending and moving, and all these things can be done. So it's bended like that, something like that. So they are pretty much flexible, and more flexible than the T4 phage, T4 phage contractile ring. Okay, and this tail is actually made up with two important stages, two important organization. One is the circular rings, and inside you are having a hollow chamber, right? So if you look at the discovery of the Bacteriophage lambda, it was by Esther Lederberg, and here is Joshua Lederberg. So actually regarding this lambda biology and lambda molecular biology, these two person are providing most of the important what you can say experimental results. Okay, so if we look at here how they have proved that there is a bacteriophage, which is a virus which can enter and destroy the host bacterial cell. So what they have done that firstly they take the donor cell, which is E. coli K12. Now they took this E. coli, and what they put there, they also put another recipient cell which is also E. coli, right? So E. coli and E. coli, and what they allow here in this case, they allow that these two cells to be conjugated with each other by the pilus, right? So after the conjugation, what they found that this new recipient cell simply get lysed. Okay, previously the donor cell is not getting lysed, but now after this conjugation, this recipient cell is getting lysed. Now in second experiment, what they have done, that they take only donor cells, and what they put here, they put little bit UV there. After some time, what they found, that they are again getting lysed. But when they put this donor cell as it is, without the treatment of UV or without any kind of conjugation, this donor cell remains as it is. It is not being dead. But now if we put some other recipient cell, and after the conjugation it transfers, that means it transfers something to this recipient cell, which causes the recipient cell to die by the lysis. On the other hand, when you put UV radiation or UV radiation to this donor E. coli cell, this UV is triggering something inside the E. coli cell, which is also telling the cell to die by the lysis, right? So that means there is something inside this donor cell which is a kind of dormant, for certain time. When we put UV, it is getting activated, and it is getting that's why it is getting killed by lysis. Or sometimes when they are transferring via the conjugation tube between this donor to recipient bacteria, in that case it is also providing that that factor into this recipient cell, so that the recipient cell is can be dead, right? So that's the reason, that there is something inside this donor cell. And later it was found that this thing which is present in donor cell is definitely a part of some foreign particle, which is called the virus, we all know. And this virus is called the bacteria eating virus or bacterializing virus. It is called bacteriophage. Okay. Now among them, they term this name as lambda or lambda phage in this case. Okay, now both these lysis can be studied under the microscope, and when they found this under the microscope, they found that there is something important particle that is kind of disrupting it, right? Now for the dormant state, in case of this donor E. coli, it is due to some kind of incorporation of the viral particle or the viral genetic material with the host. And nowadays we know that this dormant living of the phage particle inside the E. coli cell or inside the host cell is termed as lysogenic cycle, right? We know that it is termed as lysogenic cycle. And when they have encountered some stress, like in the form of UV in this case, this lysogenic cycle can be converted into the lytic phage, and those phage will lyse the cell, right? So after the cell lysis, usually how the experiments are conducted, that they take the plate and they put the bacterial on there, and then put this phage culture. You can see where the phage is lysing the bacterial cell, the cells are kind of, they are dead, and they form this kind of transparent regions which are called plaques. Okay, so this is called plaques or plaque forming units or PFUs. Now life cycle of temperate bacteriophage. Okay. Now temperate bacteriophage means in this kind of bacteriophage are having both the type of life cycle, either lytic or lysogenic, both mode of life cycle here. Now in lytic cycle, what their actual goal is, that the bacterial actual phage goal is to replicate inside the host cell and then lyse the host cell and come up with all of their progeny viruses, right? But in case of lysogenic cycle, what their aim is to put their genetic or genome, put their viral genome inside somewhere inside the host genome or the bacterial genome for certain period of time. After that, when they turn the environment is turned opposite to them, then this lysogen will be converted to the lytic phage, and then they again lyse the cell and then can come out, right? So if they want to stay for longer period of time inside the host cell, they will choose lysogenic cycle. If they want to just replicate inside the host cell, they will choose lytic replication cycle. Okay, now there is a switch, very important molecular switch to select from lytic or lysogenic cycle between their living. Okay, and we'll be seeing what are those switches to find that. Okay, but usually what happens, say this virus enters and it delivers their genome. Usually the genome for lambda is DNA. Now the DNA is then getting transcribed, then replicated into proteins, then they finally organize the proteins, and then the phage particles will come out by the lytic cycle, right? But for the lysogenic cycle, they can incorporate their genome with the host bacterial DNA or bacterial genome, and it can stay there for several times. Okay. Now one important thing I must tell you is that nowadays most of them, in fact all of them, phage particle or lambda phage particle we are working with are a variety called Lambda papa. Now what is Lambda papa here? Now Lambda papa is simply derived from two important individual lambda type, which one is Lambda Paris, another one is Lambda Pasadena. uh, the name suggests all that lambda is from this Paris or lambda phage obtained in Paris and the lambda phage obtained in Pasadena, and they cross these two and they finally produce this lambda papa, which is designated as a model lambda phage to study rest of the processes nowadays. Okay, okay, where is it? Okay, so let's move on. Now let's talk about the structure of the genome of lambda phage. If we look at the structure of the genome of lambda phage, it is having a slight unique feature, which is having the cohesive ends at both the ends. So cohesive end means simply it's a kind of it's a kind of what you know, complementary structure. It's a complementary structure or complementary sequence which can pair with each other, right? As you can see here, here is a C, here is a G, so again C, G, C, G. So C, G are the complementary of each other, then AT is a complementary, GC is the complementary. So as they are having complementary sequences from opposite direction, there is a chance that they can combine with themselves via the hydrogen bonding between the complementary bases, right? And they are having this kind of 12 nucleotide stretch of these complementary regions, both the ends, which are called the cohesive ends or complementary sites. Okay, now this complementary site is helping them to be circularized once they are injected inside the host cell. Now why it is required? Because inside the host cell, if the lambda injects their DNA as linear DNA, which is a common form of lambda DNA, if they inject it in linear form, it is having a chance to be degraded by the host cell exonuclease from the terminal portions. But if it is getting coiled in circular form, none of the exonuclease enzyme can break this DNA, right? So that's the advantage part of that. Now if we look at the lambda genome in detail, what it will look like, it will look something like this. It is having several different gene arrangement, having different sections. Like this particular gene arrangement is called it is responsible for synthesizing head, and this particular stage is responsible for synthesizing the tail portion of the lambda. And then we are having DNA synthesis regions, lysis regions, or the regions which are required for the lytic phase to be occurred, regions for responsible for the lysogenic phase to be occurred, and then rest of the parts. So these are producing the proteins which are required for the lysis of the host cell and all these things. Okay, now among all of these different arrangement, the most important part excuse me, the most important part of this lambda genome lies between this particular operon system here. And here you can see the promoter, the operator, and there are the different genes like CRO, C1. These are the most important regions, CRO and C1. These are the products that are most important, not actually products, these are the genes that are most important for controlling between the lytic and lysogenic cycle of lambda genome. Okay, if we look at here, and there is also N protein, C3, C2, and all the proteins, and another two important proteins, red and gam, that is also present here. Okay, now in this case what we can see that uh, in other video you can find in my YouTube channel, you are going to see how this switching between the lytic and lysogenic cycle usually made or designated by lambda. So I'm not going to talk about them in detail here. So just know that these are the section and this is the operon that is required, which can be obtained in two different ways. As you can see, two promoters there, promoter left PL or promoter right PR. From this promoter, it will transcribe these right-handed genes in this direction, in clockwise direction, and from the promoter left, it is synthesizing or transcribing the mRNAs from this anti-clockwise direction. Okay. Now synthesis of immediate early genes, which is important here. If we look at the synthesis of immediate early genes, the genes which are synthesizing at very beginning. Now there are three major stages of synthesis of proteins. One is immediate early proteins. Second one is the early proteins. Third one is the late proteins. Okay, so these are the three proteins. The immediate early proteins are those proteins which are required for the replication of the phage DNA. Early proteins are responsible for the production of transcription factors and transcription of this DNA of phage, and late proteins are those one which are responsible for the production of the structural portion of the viruses. Okay, so in the very beginning, in the immediate early gene, we are having N and CRO proteins. Now CRO is required to choose between the lytic or lysogenic cycle. That's why CRO is very important in this particular stage. Okay, Now, synthesis of delayed early genes. In delayed early genes, there are many more genes like C2, C3, N, CRO, O, P, and Q, and rest of this part. But majorly CRO, uh, N, C2, and C3. Now, these particular genes are required for the future times to control the transcription, to control the transcription. Okay, control the transcription of gene present inside the viral genome. So now let's talk a little bit about the lytic cycle and how the lytic cycle actually proceeds. Now the mode of action of the crow repressor is very important in controlling the lytic cycle, right? So what is happening in this picture? Try to figure out, look carefully in this picture. That the crow repressor binds to the OR3 or operator right 3. Actually, there are three operators in the right region. Operator 1, 2, 3. R1, 2, 3 is given here. So you can see at first the crow repressor binds to the operator R3, and usually the crow repressor is active only when the crow is in the dimer form. So you can find in all these cases the crow is present in the dimer arrangement. Okay, and this thing is called the RNA polymer. This is designated here. Okay, so first this crow repressor or crow dimer binds to the OR3 and it stops the transcription from this PRM through the transcription from PR continues in this case. So it will block the transcription from this particular transcription is blocked, but the transcription at this right hand direction still can go on. Okay, and this kind of transcription allows this bacteria uh, to be inserted inside the lytic pathway of their life cycle. Okay, now when the concentration of the crow repressor kind of increasing, right? Now as you can see in this right hand side there is a gene called crow. So crow is kind of controlling its own synthesis. Now as it is turning on the genes or the synthesis of gene products or mRNAs in this right hand direction, it is also enhancing the synthesis of crow proteins. So more synthesis here will be done, and here it is kind of positive feedback loop which is controlling the production of many crow due to the presence of crow dimer here in this OR3. Now as the crow proteins start to increase there, the concentration of crow proteins start to increase, the crow protein can go and furthermore they will start to bind in this OR2 region, right? Now this leads to the turning down of the transcription for PR. So as a result of that, the transcription at this direction start to halt. Now again, when the crow protein reaches a particular concentration and it is kind of binding with OR2, it is telling that no further crow protein is needed. So it is shutting its own transcription down, right? So it is kind of again controlling its own transcription, and here via negative feedback loop. So what we can see here, crow is acting as a master controller of its own transcription via positive as well as negative feedback, right? Now at much higher concentration of crow protein, a third dimer can also bind with this OR1 region here. Okay, and due to this all effectiveness, it will block the synthesis of left hand side genes. As a result, the virus actually insert into the lytic cycle of their life cycle. Okay, now synthesis of proteins necessary for the replication of lambda genome and formation of head and tail, and especially the proteins that are present here for the head protein region, and this is for the tail, and all of the genes that are present here, remember all of the proteins are termed as the name of the alphabetical order A B C D E F G H I J K L M N O P Q R, and all these different order proteins are present here. Now all these proteins are responsible for producing, some of them are responsible for producing head, some of them are responsible for producing tail. Okay, but then after producing head and tail, it is a very important job to assemble this head and tail, and finally what we prepare our mature phage. Now before that, what we can produce, if the phage already been produced their own genetic material or DNA via the replication process, then we take this DNA uh, out from the host cell and start to package it inside this phage head. Okay, that's how the everything is done here. Now the replication of the lambda genome, how the lambda genome replication is actually carried out. Now, usually the replication of lambda genome can be carried in two different ways. One is the theta mode of replication, as you can see here in this picture. Another one is the rolling circle replication, something like that, which you can see in this picture, right? Now in this case it is very important to switch between this theta mode of replication and then the rolling circle mode of replication. Now usually it's a general process of lambda genome to carry out the theta mode of replication after taking entry inside the host cell. For example, here using the cos site, it forms a circular DNA. Now once it forms a circular DNA, from the origin of replication site, the transcription begins and it starts to produce the RNA, right? Now as it is start producing RNAs, it is also being translated into a product called gene O product. Now the gene O that is present there, it is responsible for helping in the theta mode of replication. So the gene O product will come and it will bind to a certain region of the DNA and it starts to make a kind of theta-like structure inside the DNA. So as it is making the theta-like structure via the process called theta mode of replication, theta replication is done. After the theta replication, it can make a copy of its own circular DNA. This is a general way of replication of the lambda phage. But now when it requires to process all the genome inside the host virus, inside the progeny virus, to carry out the lytic phage or lytic cycle, it needs to shift the replication scheme from this theta mode of replication to the rolling circle mode of replication, right? So theta to rolling circle it for the lytic cycle, lytic phase. So for lytic phase, it requires to shift from theta to rolling circle mode. Now in this case, once it is duplicating its circular DNA, then it later uses the GAM and other products. Like GAM inhibits the REC-BC protein. So as it is inhibiting the REC-BC, finally it will block the synthesis pathway via the theta mode. It will start the rolling circle mode of replication. Now as the rolling circle mode of, usually what is going on, REC-BC is the important protein which is blocking the rolling circle mode of replication and it is allowing the theta mode of replication, right? Now as the GAM is expressing during this lytic phase, due to the binding of CRO, remember, due to the binding of CRO here, it is, sorry, where is it? Due to the binding of CRO, it is in enhancing the genes that are present in right hand side to be expressed more, and there is a gene called GAM that is present in this right hand side. So as it is expressing here, so this GAM will go, it will block the effectivity of REC-BC. So as it is blocking the activity of REC-BC, it is telling that yes, you can go from theta mode to the rolling circle mode, because previously REC-BC was blocking the theta to rolling circle mode conversion. Now as the REC-BC is getting cleaved or it is getting blocked, now blocked by GAM, now it can convert itself into the rolling circle mode. So rolling circle mode of trans replication goes on, and as it is going on, it is providing and creating more and more copies of the same gene. So what it is providing actually, so if I look at here, it is actually providing, so let me watch this, yeah, so where is it? So it is actually providing us kind of, so let me, it is kind of replicating, rolling like that. And what it is providing, let's say the genome of the phage is actually from this to this section, and it is having a kind of, so it is making more copies of the same gene, and at the both the terminal, what they are having an important site called the cos site. Remember, cos site is start to form in both the terminal, so that after the production of this many copy of the gene in same stretch of the genome, which is called a concatimer, you know that it is called the concatimer. So concatimer usually forms here. After the formation of this concatimer, what they will do, so concatimer means we have a long stretch of DNA and same type of genome is repeating, and both the terminal we are having cos sites here like that. So cos here, cos here. So cos sites are present both the terminals. So the packaging will begin. Head will be there, and they start to package section of this gene by cutting or cleaving this gene out, and inside this phage head, and what he produce here is a mature phage, mature lambda, right? So that's how the mature lambda is kind of generated here. Okay, and during this packaging, which is very important, it requires then first is to assemble this head particle, right? And we know there are protein regions present to produce this head particles in the lambda genome. So before the production of these head particles, what we are having here, we are having first 42 kD head subunit, and then using this common host cell GROEL and GROES system, this 42 kD head subunit proteins are kind of arranged together to produce what is called a shell assembly, and it is called the prohead 1, which is a very crude form of the phage head. Now during this time they require much more proteins, and furthermore cleavage to finally generate Prophage 2, which is slightly modified version of Prophage 1. Now from this Prophage 2, this is the time where this head is kind of coming to the contact of concatemer, uh, and the packaging and the maturation of head is kind of going on simultaneously, right? The packaging is kind of going on with enzymes, terminus enzymes, and this Prophage is kind of matured. So dsDNA is kind of entered inside the head. This is very very important, that before the fully maturation of the phage head, it requires to insert the gene segment or the genome segment of phage inside the head, right? So DNA is kind of injected there. After the injection, the head is kind of matured, and then the cross-linking of head is being conducted, and the head is finally made with head 2. Okay, so that's how the whole process can be done. Okay, now in this case, if the double-stranded DNA is not injected inside the Prophage 2, the Prophage is kind of disassembled and dissociated in this case. Okay, so that's very very important, that the dsDNA is kind of inserted inside this Prophage during this maturation. Okay, now let's talk about the packaging of lambda genome inside the phage in much more detail. As you can see here, this is the section. This is the section at the terminal. We are having cos sites, and we are having this Prophage coming in. So Prophage is kind of coming in, and the protein called GPF1 is also there, and cos sites are present at both the terminus, as you can see in this picture. So cos is here, and then it requires NU1 protein. Now NU1 protein is kind of guide protein. Let me write. It's a kind of guide protein which is required during the packaging of the phage genome inside the phage head. Okay, so it's kind of bound with this head and the DNA packaging regions, giving the guidance to this phage genome to be inserted inside the phage head. Okay, and at the terminal there are proteins called Q and B that are being attached there. Then as they start the packaging, and for the packaging they require energy. The energy is supplied from ATP hydrolysis. So ATP is getting hydrolyzed, and they start to package it, and while packaging, you can see here clearly that the Prophage is also matured into a mature head. So it's a Prophage previously, Prophage of phage. Now it's a matured head here. So maturation of head is also going on simultaneously during this packaging. But the packaging initiation is important to further modify the Prophage into the mature head, right? So as this packaging is going on, ATP is required, and hydrolysis is providing the energy. After a certain time, when it reaches the terminal of the cos site, it will be cleaved there due to the presence of the protein GPD, and also the other proteins like GPW, GPF2, and other tail proteins will be there. They will come and attach to this region, and finally they will provide the mature phage progeny particles. Okay, but this kind of packaging will continue from one cos site to another cos site in both the terminals. Okay, that's how they can usually know how to package and which is the amount of DNA to be packaged. Here comes the bacteriophage tail assembly pathway. So we know here it is the very beginning of the tail, which is immature part. Only GP, or they are always called GP or glycoproteins actually. So GP, and this is called GPJ, GPI, KL, and MP, and all these different proteins are there. So three GPJ proteins first assemble to provide this kind of amateur structure. Then GPI, GPL, GPK, and all of this kind start to arrange together to finally make a kind of tail structure like that. It is first called the U minus tail or U minus tail. Now then what they add? They add the GPU protein, which is important, and after adding the GPU protein they produce the tail called Z minus tail. Now then they add GPZ, then produce what you call a matured tail. So we cannot call a phage tail matured until and unless it is having two important proteins. One is the U proteins, another one is the Z proteins. Now usually U, Z, and V, both of these proteins are important. V is required for the virulence factors, and U is required to control that and other things. So to understand rest of this protein function, you can find small video in my YouTube channel regarding this bacteriophage entry to release, bacteriophage lambda entry to release in my YouTube animated video. You can find it and you can get the answer. Okay, so once they have arranged this GPU, GPZ, and finally produce a mature tail, now the tail will be directly assembled with the head particle of the phage, and the tail and the head particle of the phage assembly will take place simply via random process. So tail and head will be attached randomly via the Brownian motion theory. They just attach there, and then the fully matured phage is ready to infect, ready to be lysed outside. Okay, so this is about the lytic phase. Now let's talk about a little bit about the lysogenic cycle. So what is the mode of action for the lambda lysogenic cycle in case of lambda repressor in case of lysogenic cycle? Now in this case, the repressor binds to the OR1 because of its higher affinity, right, to this sequence. Usually the repressor is having higher affinity to OR1, so that is very very important foundation. Actually, I haven't talked about that. Usually the repressor or crow repressor usually binds with OR1 with much more affinity, then a little lesser affinity with OR2, then a little less affinity with OR3. Right? That's how the affinity is going on. Now, as at the very beginning, uh, the repressor is present in very few concentration, it first binds with the OR1 region because that is having the highest affinity region. So it binds to the OR1 section here with a higher affinity. Now then the interaction with the OR1 and OL1 blocks the transcription from PR and PL, but not from PRM. So it usually blocks the transcription in this direction. So simply it blocks it here in the opposite direction, which is required to produce the proteins for lytic cycle. And that's very logical because we don't want to turn it into the lytic cycle, we want lysogenic cycle. So it will block all the necessary genes that are required for the transcription of the protein products required in the lytic cycle. It will promote all the other proteins required to finally turn into the lysogenic cycle, right? So they will start the promoter PRM here. So now the polymerase will come and bind to the promoter PRM and will start transcribing genes present in the left hand direction in this picture. Now when more repressor is available, a second dimer binds to the OR2 cooperatively, right? So as it binds to the OR2 cooperatively, this results in enhanced transcription from PRM by repressor interacting with the RNA polymerase. Now the repressor is kind of interacting with the RNA polymerase. As a result of this interaction that is visualized by this image here, the RNA polymerase is kind of enhanced to act properly here. So here are the repressor first bind to OR1. Now due to the slight more concentration, more repressor coming and bind to the OR2, and then they will be attached to the RNA polymerase and telling RNA polymerase to act pretty finely and properly. And the interaction of OR1 or OR2 with these crow proteins, what they require here, they require the presence of another important protein called C1. Now this C1 and C2, both of these proteins are required to enhance the activity of this repressor there to finally turn into the lysogenic cycle. So then it will be bound to the RNA polymerase. It will tell the RNA polymerase to move on and synthesizing those proteins required for the lysogen conversion into the lysogenic cycle. Okay, so here comes the C2 protein that also enhances this kind of transcription. C1 protein also enhances the transcription. In both the way, it enhances the transcription and then enhances the transcription of the genes called the regions called ZIX and INT, which is required finally to INT is the gene which is required for the integration of this phage genome with the host cell genome, and XIS is a sequence which is required to release or cut out this phage genome from the host cell after the lysogeny has been accomplished. Okay, so let's look at here. So once everything is kind of going on, and there are site called ATTP. Remember in this picture also we have found the site ATTP, and two types of site is there, ATTP and ATTB. Now ATTB is present in the bacterial chromosome, that's why it's called that P. ATTP is present in the phage chromosome, that's why I call the phage or ATTP. So in the ATTPB, we are having this kind of sequence. We can see it's a kind of same stretch of sequence in both the case, in case of bacteria as well as virus. So they are having a same type of structure. So recombination event is very much possible between this lambda chromosome with the bacterial chromosome. So here it goes. It is a kind of attached via this BB with PP is kind of recombination there. After the recombination is established, this phage genome is kind of incorporated to this host cell or bacterial genome. And now the stretch of phage genome embedded inside the bacterial genome is termed as prophage. Many students find this prophage term confusing, but prophage means a section of the phage genome which is being inserted inside the host bacterial cell genome. Okay, now as it is producing this prophage, as you can see at both the terminal it is having P, B, P prime and PB prime, and then the rest of the genes for bacteria is present in both the directions. Now it is called the lysogenic chromosome. Okay, so after they form this lysogenic chromosome, it is very important to finally turn it into the lytic phage, because we know these are temperate phage. They can convert their strategy from lysogenic cycle to lytic cycle in future. So how they came to do that, so let's talk about that. Now what we know about here, that high concentration of repressor, when we having more and more crow there, right? So dimers start to bind an OR3. As it start to bind this OR3, they're kind of switch off the transcription from this PRM, right? And due to the overlapping sequence with the RNA polymerase binding site, this will not allow the polymerase to further bind with this PRM. So the transcription previously blocked in this direction, now it is being blocked in left hand side also, right? So the transcription is blocked in both the ways, and the lysogenic cycle will be repressed in this condition. And the only way to repress it by the higher repressor concentration, because this is the only way that the repressor or crow dimer will bind to the OR1 or OR3, sorry, because OR3 or OLC, because this third operator region is the most it is having the least affinity towards the binding of crow proteins. Okay, now let's talk about the prophase induction. Now this particular phenomena is termed as, it is simply the release of the lysogen from the lysogenic phage or lysogenic condition, right? So if we look at, this is the lysogen, and inside this, somewhere in this bacterial genome, there is the incorporation of our phage genome, right? And we know that sometimes in future, when they find the environment is difficult to stay in, and the bacteria is kind of in tough situation, it can die. So in this case, the phage genome start to emerge as again lytic phage, and then start lysing the bacterial cell and can come out. So how to revert the lysogenic cycle into lytic cycle? So lysogenic to lytic, in this case it is called the phage or prophage induction. So in this case, what they require any kind of stress. Now what kind of stress here? The stress here can act as DNA damage due to any kind of irradiation like UV radiation, or any kind of chemical stress, something like that. Due to this kind of stress, when the DNA of the bacteria is turned into the single stranded DNA, and it also starts producing or expression of some proteins called rec proteins, right, which is required for the repair mechanism of the DNA repair mechanism of bacteria. So as the rec proteins start to emerge there, that the rec protein will come, and they are kind of degrade this repressor that are present there. So they are kind of block or cleave all the crow repressors that are bound to OR1, 2 and 3 in all this direction. So as they are cleaving this repressor in these particular stages, they are kind of triggering this prophage to finally come out. How? Because they kind of inhibiting this crow, and all the regulation from controlling between lytic and lysogeny is kind of open up now, and then especially they start expressing the genes that are present in left direction as well as in right direction. Both the cases the transcription is kind of on, because there is no crow protein to regulate that, right? Because there is the crow is the only master protein to control it, right? Now in this case, as the left region or the transcription is going on from the left promoter region, it is providing us XIS regions or proteins which are required for the excision of this phage genome from the bacterial genome. How? So here it is, again the process will be done via the reverse recombination stage. So again, if they kind of form bulge like that, and from this B prime to B and P prime to P, the crossover will be done, and the phage genome will come out. And as the phage genome will come out, then they will start producing more and more. Remember, they will start producing as they are also transcribing from the right promoter region. They will start producing more crow here. So as they will produce more and more crow here, the crow will bind to the OL1, 2 and C, and then enhance the transcription in the right hand side, blocks the transcription in left hand side. So it will convert it into the lytic cycle here, and lytic cycle will be done, lysis of the cell will be established, and then the phage particles can come out. Okay, now the question remains, when the lambda executes its two life cycle, lytic or lysogenic, how to figure it out, right? Now usually it depends on two major factors. One is the nutritional status of the host cell, and second one is the multiplicity of infection, or MOI. Now in the first thing, which is nutritional status of the host cell, the nutrient rich medium. If there is a nutrient rich medium and the high concentration of FTSH protease present there, it can cleave the C2 protein, and then finally it can turn into the lytic phase, right? For example, if we present it in the nutrient poor medium, there will be low concentration of FTSH protease, and in this case the accumulation of C2 protein start to emerge and it will convert into the lysogenic cycle. So more nutrition, lytic cycle; less nutrition, lysogenic cycle. Second thing is the multiplicity of infection. Now, if there is a high multiplicity of infection, it will be lysogenic cycle. If it is low multiplicity of infection, it will be lytic cycle. Now the multiplicity of infection is termed as the number of infectious virus particles divided by the total number of target cells present in a defined space. Now if the total number of target cells present in the defined space is very less, in those cases they always try to convert into the lysogenic cycle. But if the Phage particles are they are less than the host cell, usually they follow the lytic cycle, because that's the strategy. If there is less bacteria and more Phage, and if they start to kill all the bacteria, then they won't be having any host cell to survive, because bacteria always require host cell for their living, right? So they never want the host cell to be degraded all the time. So if there is adequate number of host cells, only in those cases they will kill the host cell via the lytic cycle, right? That's the multiplicity of infection is telling us. Okay, so that's it, and I hope that's helping you. Thank you.